Hello students, how are you today? I hope you are fine. Today there is turn for the second lecture during our course. As you remember, the previous one was about PCR reaction. You already have basics about this technique, so we can continue with the lecture on real-time PCR reaction. Real-time PCR is very precise and sophisticated modification of classical PCR assay. Traditional detection of PCR products with electrophoresis was replaced by their detection during the reaction, and that's why we call it real-time PCR. Please look at this picture. Here we have our sample. This is sample, yeah, from which DNA has to be isolated. As you remember from previous lecture, PCR is used to amplify specific fragment of DNA in huge number of the copies. This fragment is visible here as green line. Here you have many fragments of DNA, but we are interested in only this specific one, marked as green. This will be our product. So what happens during PCR reaction? This is thermocycler, and after PCR reaction, after 30 to 40 cycles, we have a lot of copies of our fragment. Here, from one fragment, we had huge number of the copies of the same fragment. And at the end, we have to conduct electrophoresis to check the result. So this is result. As you remember, we will see flashing band, which represents our fragment amplified in huge number of the copies. And this is the end of classical PCR reaction. If we see one flashing band, the result was positive, and that's all. We can tell the DNA fragment was detected in research sample. Real-time PCR is much more precise and sensitive. You can observe your reaction on the screen of computer connected with special thermocycler in real time. It means that you see what happens with your sample at this moment. That's all. It's great, isn't it? That's why we'll call it real-time PCR. How is it possible? How we can observe our reaction at its duration? How is it possible? You have to use special reagents which allow detection of PCR product already during the early stages of reaction. In traditional PCR, we have to use agarose gels to detect the reaction products obtained in the final stages of PCR reaction, and that's why we call it endpoint PCR. So traditional PCR reaction is also called endpoint PCR. In real time, you don't have to conduct any electrophoresis. You will get results on computer screen. That's all. To understand how PCR works, it is necessary to know something more about three phases of reaction. Please remember, phases of PCR are not PCR cycle steps. We are talking about phases of whole reaction, not single cycle. So these phases are called, we have three phases: exponential, linear, and plateau. Exponential phase. In this phase, there is doubling the amount of product in each cycle, assuming that we have 100% reaction efficiency. The reaction in this phase is highly specific and precise. The next phase is called linear. Primers and nucleotides are gradually used during reaction. Activity of DNA polymerase is lower. Reaction slows down and the products are gradually degraded. And the last one, plateau phase. For classical PCR, in this phase we detect the product on agarose gel. In this reaction, In this phase, the reaction is completed. There are no new copies of a product, and amplicons degrade after an extended period of time. So please remember, PCR reaction, the whole reaction, can be divided into three phases: exponential, linear, and plateau. And the most important for real-time PCR reaction, the first phases. The first phase is the most important, exponential. Exponential phase is most important for our reaction. So what are the basics of real-time PCR? Technique has various names and abbreviations. We call it quantitative PCR, real-time PCR, or just qPCR. Q means quantitative. So the most common abbreviation which you can find in the internet and in the scientific papers will be qPCR. But please remember, it should not be called RT-PCR because RT abbreviation is reserved for reverse transcription reaction. RT does not mean real time. RT is reverse transcription. So what are the reagents we have to use to make reaction mixture for real-time PCR? We need just the same reagents as for conventional PCR reaction. It means DNA sample, nucleotides, DNTPs, primers, and also enzyme, DNA polymerase. But we have to add some additional component to the mixture. And this component may be special dye intercalating into DNA, or instead of it, we can add molecular probe specific for the amplified sequence. The reaction is conducted in a special thermocycler and observed on the computer screen. Real-time PCR enables the evaluation of the quantity, the number of copies or concentration of the product amplified during reaction. That's why real-time PCR is quantitative technique, while classical PCR is qualitative. Please remember that. Real-time is always quantitative. It means quantitation. What is quantitation? It is estimation of the number of the copies or concentration of the product in our sample, and classical PCR reaction is qualitative. Qualitative means that we can only say that we can only have negative or positive results, plus, minus, 1 or 0. We cannot quantify the amount of the DNA in classical PCR. We can do this using only real-time PCR. So real-time is quantitative. How does it work? In real-time PCR reaction, special detector measures the light. The light is called fluorescence and is emitted by the products during the reaction. This way we can monitor an amplicon production after each PCR cycle in real time. So once again, we can calculate the quantity of desired fragment of DNA at the beginning of the reaction, which is impossible in the conventional PCR technique. Do you remember the phases of PCR? Here you can see them once again. In the first cycles, the level of fluorescence is very low. Then the reaction reaches exponential phase. This fragment is exponential phase. Later, followed by linear phase. This fragment of the reaction is linear phase. And the last one is plateau phase here. The reaction ends and we have no more products during the reaction. This is plateau phase. The most important for quantification of the results is exponential phase. the first one. This is the most important for quantification of DNA in our sample. Exponential phase only. What are the advantages, the positives of real-time PCR? This is full monitoring of the reaction. Short reaction time. We can finish the whole reaction even in half an hour, but most popular equipment, thermocyclers, can finish the reaction within about 2 hours. Very high sensitivity. Real-time PCR reaction is about 10,000 times more sensitive than classical PCR reaction. 10,000 more sensitive. You don't have to make electrophoresis at the end of PCR reaction. Analysis of the melting temperature of products provides confirmation of reaction specificity. This is the special type of analysis about which I will talk to you later about during this lecture. Another positive, another advantage, wide range of measured concentrations. So we can detect one molecule of DNA, one fragment of DNA, up to 10 billions of the copies. So very wide range of concentrations which can be measured by real-time PCR. And this is highly sequence specific. It means that we can detect mutations in the DNA. So we can detect the change of the sequence in our fragment. We can detect the mutations in our research DNA. So what is the equipment needed to conduct our reaction? The thermocycler, and of course the most important part of this machine is heating block. And this is element which gives appropriate temperature in every step of each PCR cycle. It heats or cools our tubes during the reaction. The next one, fluorescence signal may be initiated with. So we need the source of energy which is needed to excitate the fluorescence emitted by the products. And we can use it we can achieve this energy by the use of the lamps. We can use the halogen or xenon lamps, the lasers, or LED. And the last part of the equipment is fluorescence detection. So we have to get the fluorescence, we have to excitate the fluorescence, and then we have to measure it. And we measure it with the special fluorometer, which is connected with the thermocycler. So on this slide you can see the examples of various real-time PCR systems. So for example, we have many producers of the real-time systems. This one is Bio-Rad CFX96. This is Eppendorf Realplex, Chromophore, Stratagene, and Light Cycler 480 produced by Roche. And this equipment, the same one, is in our laboratory. So we use this type of the machine, Roche. But of course, many other producers also offer their systems on the market. So reaction is conducted in special plates with wells. This is the plate, and you see the wells. The holes in the plate are called wells. And we have two formats of plates, 96, this one, and 384. So we have 96 wells or 184 wells. We can choose which format is better for our applications. On this plate, we have 4 times more wells than on this one. So we can conduct 4 times more analysis than on this one at the same time. And below you see the heating block of the machine, which creates the temperature for the reaction. So after preparation of the plate, we have to put the plate inside the machine and reaction can be started. So the first thing to do is to prepare the reaction mixture, prepare your samples, and put the reaction mixture and the samples with the pipette into separate walls of the plate. Then you have to cover the plate with the special optical foil. Then you have to centrifuge the samples, and finally you have to put your plate on special tray of the machine, and then you have to press this gray button, and after pressing this button the tray goes inside and you can start the reaction. Fluorescence of each sample is proportional to the amount of the amplified product. It means that the stronger the fluorescence signal is, also the more copies of product we have. So the stronger signal of the fluorescence, the more copies are present in our sample. So we can monitor the whole reaction by measuring the fluorescence of samples after each cycle of the reaction. There are several methods for detection of increase in amount of PCR product. All are based on fluorescent compounds. They require special thermocycler equipped with fluorometer, measuring fluorescence emitted during reaction. Our fluorescent compounds are also called fluorochromes. So fluorochromes have to be excited by light of a specific wavelength, and emitted energy must be detected and measured. As you remember, we can excite fluorochromes by 3 sources: lamps, LED or lasers. Lamps emit light with a wide wavelength range, while the LED or lasers narrow. Detectors may have filters through which only one wavelength may be detected and measured. So, what chemistries we can use during real-time PCR reaction? We have two groups of chemistries, two groups. The first one is intercalating dyes, and the example of this group is cybergreen 1. This is the most popular fluorescent dye, cybergreen 1. This is the first group, and the second group are fluorescent dyes in probes. So cybergreen emits strong fluorescence signal after binding to double-stranded DNA fragment. And probes, we have many types of the probes, and in this presentation you will know two types of probes: Tachman and molecular beacon. So what is a probe? The probe is a single-stranded DNA fragment with a specific base sequence. And this sequence is labeled with fluorescent dyes. One is reporter and the second is quencher. Probe has its own sequence and is used for specific detection of complementary sequence of target DNA amplified during PCR reaction. Now, when you know what chemistry can be used in real-time PCR, we can divide real-time PCR technique into two groups of detection. We have non-specific detection and specific detection. The criterion of division to non-specific and specific group is the type of fluorescent dye used in reaction for DNA detection and quantification. So it will be cyber green or probes. The first group, non-specific detection. Fluorescence in this group is generated by special intercalating dye. The most popular is cyber green 1. It emits a strong fluorescent signal after binding to double-stranded DNA. We also call it dsDNA. It means double-stranded DNA. So we obtained strong signal emitted by cyber green only after hybridization with DNA. So how cyber green works? On this slide you see 3 steps of PCR cycle: denaturation, annealing and elongation, and what happens in every step. So please look at denaturation. We have denaturated DNA strand, so this is single-stranded DNA, ssDNA, and F means fluorochrome. In this case, F is cyber green. So these are molecules of cyber green. And now they are free, and we have no connection between cyber green and DNA. At the beginning of amplification, the reaction mixture contains the denaturated DNA, primers, and dye. The unbound dye molecules produce only a minimal fluorescence signal. It is also called background. So we have almost no signal. The signal is very weak. So we can tell that we have no signal at this stage, in this step. So the next step is annealing. What does it mean, annealing? It is the hybridization of the primers to the single-stranded DNA. So this is our DNA, single strand, and this fragment is primer. And primer hybridizes to DNA. And first molecule of the cyber green comes between 2 strands of DNA. After annealing of the primers, a few dye molecules can bind to the double-stranded DNA. DNA binding results in emission of strong light signal by cyber green after excitation. And the last step, elongation. What happens in elongation step? Our enzyme, DNA polymerase, starts to build new strand of DNA. So it reads the old strand of DNA and synthesizes the new fragment of DNA. So our new DNA strand is longer and longer, and more molecules of cyber green can be bound to the DNA, and stronger signal can be emitted and measured by the detector. So during elongation, more and more dye molecules bind to the newly synthesized DNA. If the reaction is monitored continuously, an increase in fluorescence is viewed in real time. After denaturation of the DNA in the next heating cycle, the dye molecules are released and the fluorescence signal falls down. And it will repeat once again. So as you remember, the typical number of the cycles in the reaction is between 30 to 40. So cyber green binds to double-stranded DNA with a higher affinity than for single-stranded DNA. In result, unbound dye shows very little fluorescence. However, fluorescence is very strong, about 1000 times higher after DNA binding. This makes cyber green a sensitive indicator for the quantity of dsDNA present in the reaction mixture at any time. So please look at this simple animation and you will see how CyberGreen works. So before I start the animation, please look at the scheme. We have double-stranded DNA. We have DNA helix here, one strand, second strand. And now I will start the animation and you will see what happens with the CyberGreen. You see, once again. So you saw that before binding to the DNA, we have no light, and after binding to DNA, we see the strong fluorescent signal which can be measured by the detector. So this CyberGreen emits signal after binding to DNA. When we have no binding, we have no signal. Here we have the signal. Very simple technique. What are the advantages and disadvantages of CyberGreen? The advantages. This is cheap and easy to use reagent. It emits a strong fluorescent signal after binding to dsDNA. It does not inhibit the reaction of amplification. It does not require any fluorescent probe and does not require any particular experience in the design of the probes and is not affected by mutations in the target DNA. So these are the main advantages of this reagent, but of course it has also disadvantages. And what is the most important one? Disadvantage. As you remember, now we are talking about non-specific detection. So this dye, the CyberGreen, is non-specific dye. So it means that it binds to DNA non-specifically. So it requires reaction optimization by conducting melting curve analysis to make sure of specificity of amplicons generated during reaction. So why this is so important? Because CyberGreen can produce fluorescence from any double-stranded DNA fragment. So if you have only one fragment after reaction, the whole fluorescence will come from this fragment. But let's say that we have also additional fragments which are not specific for this reaction. So in this case, we will have more products and every product will emit fluorescence because this CyberGreen is non-specific. It means that it will bind to every double-stranded DNA fragment and it will emit the light from any fragment of DNA. That's why we have to check after reaction, after real-time PCR reaction, if we had only one specific product after reaction, and in this case our results will be okay, or maybe we had more than one product in the reaction, and in this case we'll have to make the reaction once again and to optimize the reaction to remove the unspecific products from the reaction. We want to have only one product after reaction, not additional products, only one product. So melting curve analysis. As you saw on previous slide, one of the major advantages of CyberGreen 1 is that no probes are required. However, specific and non-specific double-stranded DNA PCR products generate the same fluorescence signal after binding CyberGreen 1. To distinguish between fluorescence coming from specific and non-specific products, we have to make melting curve analysis after PCR reaction. During melting step, the sDNA product is melted into SS. SS means single-stranded DNA by increase in temperature with fluorescence reading at this time. This way we can check if our reaction gave only one specific product or more products which are non-specific. Of course, we want only one specific product of the reaction. If you have more products, reaction has to be optimized and repeated until one product will be generated. So, how it happens? This is picture representing melting curve analysis. So we have temperature which increases from 76 up to 91 Celsius degrees in this example. So temperature increases generally from 65 to 95 degrees, and cybergreen is slowly released from DNA fragments. So what we have here, we have our dsDNA fragment, and these small green dots are molecules of our cybergreen. So we have product of PCR, with which molecules of cybergreen are bound. And we increase the temperature. And what happens? So cybergreen is slowly released from DNA fragments. When fluorescence level reaches 50%, this is this level, software of PCR machine calculates the temperature of this moment. So this is this moment, and this is temperature Tm. Tm means temperature of melting. And machine calculates the temperature of this moment. It is called temperature of melting, Tm. Temperature rises up till over 90 Celsius degrees. DNA is completely denaturated and cybergreen stops emitting fluorescence. So here we have complete denaturation of the product. So at the beginning we have the DS product, and after denaturation we have SS product. So when we denaturated completely our product, our cybergreen is not longer bound to the DNA and stops emitting fluorescence. We have no fluorescence. Please look at this one. We have the level of fluorescence from 0% to 100%. So we have full fluorescence, and during the denaturation, the fluorescence starts to slow down, and when it reaches 50%, we can calculate, the software of the machine calculates Tm, and of course the temperature rises up and we have more and more molecules of our products denaturated, and less lower and lower level of fluorescence, and finally we have no fluorescence at all. So this is melting curve analysis. So if only one specific amplicon was generated during reaction, we will see one peak representing this product. So this is this peak. So we have 1, 2, 3, 4 samples, and every sample gave the same product. We see the same peak for each sample. It means that we had only one specific product. This is perfect situation. If there will be more products after reaction, additional peaks will be visible on the screen. See here? Almost all products gave one specific, almost all samples gave one specific product, but in this case, in this example, we see additional peak. This peak had lower temperature of melting, and of course it was at lower concentration. But we see in this example that we had additional product. It should not be detected in the reaction. So we want to have the situation in which all samples gave us one specific product. We don't want to have any additional unspecific products like here. I'm talking about the cyber green, but of course we can also use another types of the non-specific dyes. Not only cyber green can be used. Instead of it, we can use other dsDNA dyes like Risolite, LC green, Eva green, or Cytonine. So these are the examples of next generation dyes. So see, cyber green is second generation dye, but we have also third generation dyes, and these are Cytonine, LC green, Eva green, or Risolite. So we can choose which dye we want to use. The most popular and the oldest one is cyber green. It is still used in the laboratories, but of course we can choose other types of the dsDNA dyes, and we can choose between Risolite, LC green, Eva green, or Cytonine. So this part of the lecture was about non-specific detection, and the other group is specific detection. And I told you already that specific detection means probes. The first type of the probe is Tachman probe. Tachman probe is also called degrading probe. It is oligonucleotide, and approximate length of this probe is between 20 to 30 nucleotides, and the probe is complementary to the amplified DNA sequence. At its 5' end there is a fluorescent dye, and at the other end, 3', the quencher is attached. If the probe is not hybridized to DNA, the short distance between dye and quencher completely blocks fluorescence. So the quencher is the reagent which can block fluorescence from the reporter. During each cycle of PCR, the probe recognizes a complementary region of the template DNA and hybridizes between regions of primer annealing. During elongation step, polymerase cleaves, it means cuts, the probe, releasing the fluorophore into solution where it emits fluorescence. The amount of fluorescence during cycle is proportional to the amount of specific product present at that time. So please remember, all probes are the examples of specific detection. When you use the probe, the signal from the probe which you measure comes only from your product. Even if your reaction will be present unspecific products, you will not have the signal from them. The signal comes only from our specific products we want to detect in our reaction. So that's why probes are called specific. They emit light only from specific product from the reaction. So how our probe works. So we see once again the steps of the PCR cycle: denaturation, annealing, and extension. So during the first step, denaturation, we have once again single-stranded DNA, ssDNA, and this is the probe. This is fluorescent dye reporter, and this is quencher, reporter and quencher. And see, fluorescent reporter dye at one end is quenched by fluorescent quencher at the other end. So they are very close to each other, and quencher completely blocks the emission of the reporter. And what happens during the annealing? So we have our DNA, ssDNA template, and this is primer. This is primer, and this is enzyme, Taq polymerase. This is our probe. As you remember, this is fluorescent dye. It means reporter. This is reporter, and this is quencher. And what happens? Our enzyme starts to build the new strand, and finally it meets with the probe and starts to cut the probe from this place. And it happens during extension step. So Taq DNA polymerase separates the quencher from the reporter. So this is the reporter, this is the quencher. And finally, they are far from each other and they are separated. And quencher is not longer able to block the fluorescence. So now we can measure fluorescence because the quencher is not able to block fluorescence anymore. Okay? So when we cut the probe with the Taq polymerase, we degrade completely our probe, and the final is that we can measure the fluorescence emitted by the reporter. So this is the animation of Taqman probe in action. So before I start the animation, please look at the picture. This is our DNA strand here. This is primer, and this is Taqman probe. Reporter, quencher. Quencher blocks emission from the reporter. And what happens? Please look. This is our Taq polymerase. Once again. So polymerase starts to build the new strand of DNA from the point in which primer was hybridized. So once again, and what happens here? Our enzyme meets the probe and starts to cut the probe. Please look at this. Did you see? We had the probe here, but the polymerase met this probe and started to cut the probe into single nucleotides. And finally, what we have emission of the fluorescence from the reporter. Once again, please look at this one. Meeting of the probe with the enzyme, cutting. And finally, emission of the signal. Very simple. I hope that everyone understood how it works. And one interesting thing. So, do you know how we call it Taqman? Taq is from Taq DNA polymerase because polymerase cuts the probe during the reaction. That's why we call it Taq. And man comes from the Pac-Man. Do you remember the Pac-Man game in which this creature was used in the game, and you can use this creature for eating the ghost and other dots in the labyrinth. So I'm sure that you remember this game, Pac-Man. So that's why we see the Pac-Man, and the Pac-Man represents our enzyme. That's why we call it Tacman from Pacman. Pac is replaced with Tac and man is then added, so we have Tacman. The next type of the probes is molecular beacons. So molecular beacons have a reporter at 5' end, while quencher is at 3' end. The regions at the ends of the probe are called stem and are complementary to each other and hybridize at low temperatures to form a hairpin structure. So reporter and quencher are located close to each other and there is no emission of the signal. The inner region of the probe is called loop and is complementary to the sequence of amplified DNA fragment. At the appropriate temperature, the probe binds to the DNA, what results in separation of the reporter from quencher and the emission of light, which intensity is measured by detector. So, how molecular beacons look like? I told you we have stem sequence, this is this fragment, and the loop sequence, this is loop. So this is reporter, this is quencher. And see, this fragment is complementary. So this is one strand, one fragment, and second fragment. These fragments are complementary to each other, and that's why we have connections, we have pairs of nucleotides between these two fragments of the probe. And this is stem. And inner part of the probe is loop. This is loop. And what happens when probe finds the complementary sequence for the loop sequence here? It will open like here, and we have separation from reporter and quencher, and reporter is not longer blocked by the quencher and can emit signal which is measured by the detector. So this is closed probe, this is open probe. As you see, this is the loop sequence, this one is complementary to the fragment of the sequence of our PCR product. So this is hybridization here, and finally, as a result, we have open form of the probe and separation of the reporter from the quencher. So we can measure the signal of the fluorescence. And once again, another scheme. So denaturation, annealing, and extension, 3 steps of each PCR cycle. We have closed form of the probe. This is stem, this is loop here. And during annealing, we have our ssDNA, we have primer and the enzyme, and the loop sequence is complementary to this fragment of the product of PCR, and now we have opened form of a probe and separation of the reporter from the quencher. And when our enzyme meets with this probe, it removes the probe from this fragment, and the probe closes and forms this and stays in a closed form. So it is removed, and then the probe is closed and can be used in the next cycle of the reaction. And animation of this, of this. So once again, this is our strand of DNA, S as DNA. This is our sequence, which is complementary to the loop sequence here. And this is the stem. This is stem of the probe, and this is loop of the probe. Reporter and quencher. And this sequence is complementary to the loop sequence in the probe. And what happens? Please look. Finally, we have open form of the probe and separation of reporter from quencher, and the reporter emits strong signal which is measured by the machine. That's all. Once again, closed, open. So this is how molecular beacons work. If there is no sequence the probe can bind with, it forms the hairpin structure, and the fluorescence is blocked. So this is hairpin structure of the probe. So once again, okay. So when there is no sequence to bind with, the probe is closed and forms hairpin structure. Slight differences between the loop sequence of the probe and the sequence of amplicon, which is generated during PCR reaction, largely destabilizes the binding between probe and DNA template, and that's why we can use molecular beacon probes for mutation detection. And this is one example, SNP, single nucleotide polymorphism. This is one of the type of the mutation. So what are the applications of real-time PCR? Clinical diagnostics of microorganisms, of the viruses, which are the agents causing human diseases. Bacterial, viral, plant pathogen detection, absolute pathogen quantification, drug therapy efficacy and drug monitoring, so we can monitor if the therapy is effective or not. Differential gene expression, SNP genotyping, so this is detection of the mutation, single nucleotide polymorphism. Food pathogen testing, so we can also find eventual pathogens in the food, and plant pathogen detection. We can also detect many pathogens, fungi, bacteria and other, which cause serious diseases of the crops. And the last slide, please look how many factors can affect the efficiency of real-time PCR reaction. We can have the problem with the inhibitors, hemoglobin, urea, heparin, organic or phenolic compounds, and many others. We can use some PCR enhancers to provide better efficiency of the PCR reaction real-time. We can have the problem with DNA degradation, tissue degradation, unspecific PCR products, lab management, DNA dyes. So when DNA is degraded, it means that the quality of the DNA sample is poor. And I told you during the previous lecture on classical PCR reaction that you have to have very high quality of DNA sample. It will give you the good reaction. If you have poor quality of DNA, your reaction will be not good and not effective. DNA dyes, cycle conditions, you have to experimentally choose the best conditions for your reaction. You have to optimize the reaction. It is called reaction optimization. Optimization means choosing the best conditions for the reaction. Also, hardware, so PCR platform, the producer, the software, the machine, and so on. PCR reaction components, you can buy components from many producers. You can choose from many offers from the reagents from real-time PCR reaction. And of course, DNA concentration. When you have too much concentrated DNA in the sample, your reaction will be completely inhibited. Please remember, you cannot use highly concentrated DNA samples directly to the reaction. If you will have too much concentrated DNA, your reaction will be completely inhibited. That's why you have to measure the DNA concentration before the reaction. And if DNA sample is too much concentrated, you have to dilute your samples before starting the reaction. And this is all for today.